A very welcome back to all. Today we are going to understand the topic of methods of studying plant communities. And these methods of studying plant communities are actually important because based on these methods, the characters of community can be obtained. Right, so we now start with the understanding of these methods. So as I told that these methods are used to obtain the characters of community. Now you might be thinking that what are these characters that we are talking about? There are many characters of community, and that is actually a totally separate topic for you, and that I'll be probably taking in the next video, in the next lecture. So for now you can just remember that to obtain these characters of the community, the characters that we are talking about, we need some methods, and these methods that we are going to discuss now are utilized from time to time. So with the changing time, the methods are also changed, or sometimes a combination of 2 or 3 methods are used to obtain the characters of these communities. So now most of the studies of community ecology is done with respect to only plants, right? So whatever investigation that has been done in community ecology is mainly done with plants, and therefore the methods that we are going to talk about right now, the methods that we are going to discuss right now will also be related to plants, because though community could also be in animals, it could also be in plants, right? But most of the community ecology has been studied with plants, and so the methods are also explained with respect to plants. Now these methods that we are talking about, that is the methods that are used to study the communities, the plant communities, these are floristic methods, physiognomic methods, and phytosociological methods. So basically these 3 types of methods are used, and we are going to now understand each of these 3 methods in a bit more detail. So let us first understand what is floristic method. So these are obsolete methods. Now obsolete methods means that these are now outdated methods, these are not used anymore. In previous, maybe many years back, these methods were used to characterize the community, but now these are obsolete methods, these are not used anymore. Or maybe only this method is maybe when we are going to characterize a community, we cannot rely only on these method, right? So what is done in this method? The flora is studied by listing various genera and species present in the community. What is done in this method? That flora is studied, right? What is done? Only the listing of genera and species that are present in the community, only that listing is done. So we know what is genera, right? In the genus there are many species, right? So like all the genus are recorded, and the species that come under that genus in the community, that is listed. Now therefore this is a kind of descriptive method where vegetation is described in terms of flora. So as I told, this is a descriptive method. So what is done? Only record of the flora present at a particular place is made, right? So for example, if you find at a place maybe some flowering plants, maybe some trees, maybe some grass species, so all of these species are simply recorded and kept, right? So there may be many species, there may be many genus, and all of these are recorded. So this is the flora, and but this method, as I told, are obsolete methods, not used anymore, or this is not the only method on which we can rely to understand the characteristic of a community, because this gives no proper idea of structure, composition, growth forms, etc. of the community. So this method is not going to give us an idea of what would be the structure of the community, what is the community composed of, and what are the various growth forms that are present, right? So this is just a descriptive method based upon just listing down of the species present there and the species belonging to various genera, right? So therefore this is called floristic method, where only flora is taken into consideration and no other parameters are considered. And also this is an obsolete method, as this is not used anymore, or we cannot rely only on this method. So now we come towards the second method, which is called physiognomic method. So physiognomic methods, first we need to understand that what is physiognomy. So physiognomy is like considering the physical characters. When something is studied based upon its look or its physical character, like how does something looks like, then it is called physiognomic method, as you can relate the term physiognomy with physical characters, the outer characters of species or outer characters of the community based on that. So this method is basically based on the physical characters, the look of the community. So what is done in this method? Various species of community are studied chiefly in terms of their life forms, right, general stature and spread. So what are the life forms that we are talking about here? By now I think you all know what are Ronquier's life forms, right? So this method studies the community in terms of the Ronquier's life forms, right, the general stature, that is how does a community look like, and the spread, that is what is the spread of these communities. So all these terms are considered, and all these parameters are considered in physiognomic methods. So in the physiognomic method, the most popular method that is used is Ronquier's life form method, right? So I hope now everyone already knows Ronquier's life form method, because I had explained Ronquier's life form in the previous lecture, right? So, and if you are not clear with this, you may go back to that video and clear your concept regarding Ronquier's life form. So if you know what is Ronquier's life form, you will very easily understand physiognomic method, right? So as I told that this method actually considers the life forms, the general stature, right? So the most popular method that is used is Ronquier's life form method. So what is done in this physiognomic method? Record of different life forms is done, and secondly, biological or phytoclimatic spectrum is prepared, right? So different life forms are recorded. So what does that mean? Now we know that Ronquier had classified the plant species into various life forms. These were phanerophytes, chamephytes, cryptophytes, hemicryptophytes and tharophytes, right? So you all know now that phanerophytes are tall tree-like plant. They have their they have their buds exposed, right? Then chamephytes, then cryptophytes, right? And tharophytes are the desert plants, and they have their buds actually hidden, right? So depending upon the environmental conditions, how does the what is the position of the perinating birds? Based on that, Ronquier had classified plants into these 5 groups, right? So what is the first step in physiognomic method? That we have to record the different life forms present in an area, right? So various species are usually present in an area, so all of those species are recorded. We list down those species, right? And those species are then distributed among the different life forms, right? Say for example, in an area there is say neem trees present, right? So what I'll do, I'll list down that there is neem tree present, and I will see that it falls under which of these life forms. Okay, it falls under phanerophyte, right? So what I'll do, I'll list down the plant species which is neem, and I'll also identify that it is a phanerophyte based upon its characters, right? Now in the same area I might see that there is some cactus species present. Okay, so what I'll do, I'll list down the cactus name, and I will based upon its character identify that it is a tharophyte, and I'll list down that this cactus is a tharophyte. And like this, there could be n number of species in an area, and those species belongs to certain life form given by Ronquier, right? So some species might fall under phanerophytes, some species might fall under chamephytes, some species might fall under cryptophytes, and similarly it may fall under hemicryptophytes or tharophytes. So the first thing that is done in physiognomic method is recording of the different life forms. So you have to first list down the species present there, and based upon the characters of the species, you need to identify its life form, whether it is phanerophyte, chamophytes, tharophytes, or any of this, right? So this is the first thing that is done, and then after that we are going to make biological spectrum. Now based on the data that we have here, we are going to make the biological spectrum. Now you all must be thinking that what is biological spectrum? So the next thing that we are going to understand now is biological spectrum, and remember that biological spectrum is also a topic in your syllabus, in your unit. So you need to understand this biological spectrum well enough, right? So now the next thing that we are going to understand now is biological spectrum. So what is biological spectrum? It represents percentage of total species in a community. So what does the biological spectrum indicate? It shows that what is the total percentage of species that are present in a community. A community has what percentage of species, that is what is represented by biological spectrum, and it represents the percentage distribution of the species among the various life forms. And what is the next thing that it shows? That what is the percentage distribution of the species. Now as I told that we are going to record all the life forms that are present, so what this biological spectrum will do, it will also calculate the percentage of species in each life forms. For example, in phanerophyte, what is the percentage of species that fall under phanerophytes? Is it 5%? Is it 10%? Is it 15%? Similarly, what is the percentage of species that falls under chamephytes? So it could be maybe 5%, maybe 15%, or any percentage. So this biological spectrum gives us an idea of the percentage distribution of the species among the various life forms. And so in biological spectrum, percentage value of each life form is calculated. So the first thing that we have to do in biological spectrum is to calculate the percentage value of each life form, right? So and when we have that percentage value, we can then prepare the biological spectrum. So do not worry, we are going to understand this in a little more detail. So the first thing, let us understand this point. Percentage value of each life form is calculated. So what does that mean? That we are going to calculate the percentage value of each life form. That is, we are going to calculate the percentage value of phanerophytes, the percentage value of chemiophytes, and all the 5 life forms. So let us understand this with an example. Say for example, in a community, the total number of species that are present is 100, and the number of individual life form, say phanerophytes, is 30. This means that in a community there are 30 individuals that are of phanerophytes. Now similarly, there could be maybe 20 individuals of chemiophytes, there could be 70 individuals of therophytes, or any number could be given to different life form. And this number, how we get? We can only get this number when we list down the species present there. So we have to list down the species and identify its life form. So when we identify, so for example, we found that the number of life form that is phanerophyte is 30, which means that 30 individuals are from phanerophytes. So how are we going to calculate the percentage value? This is simple percentage formula. What we will do, that we will divide, that is number of individual life form divided by total number of species into 100. So we will get the percentage value. So what is the number of individual life form? It is 30, right? So that it is 30 divided by total number of species, right, and into 100. So okay, this is a small mistake actually. Here I had to write total number of species is 60, right? So what will happen, that number of individual life form would be 30 into 100 upon total number of species would be 60, right? So here actually it's a mistake, I had to write here 60. So when we try to calculate the percentage value, what we will get is 50% of phanerophytes. So what will happen, these all will get cut, right? It will get divided, and what will remain is 50. Right, what we will get is 50, which means that in that community there is 50%, there are 50% phanerophytes present, right? I hope now you can understand that what do I mean by percentage value of each life form. So here we calculated the percentage value of phanerophytes life form. Like this we can calculate the percentage value of all the remaining 4 life forms, which are cryptophytes, hemicryptophytes, chemiophytes, and therophytes. So when we get this percentage value, then we will prepare the biological spectrum of that area. And when the biological spectrum of that area is prepared, it has to be compared with normal biological spectrum, which is given by Ronquier. We are going to understand this in the next slide, that what is the normal biological spectrum given by Ronquier, right? That is also called the physiognomic flora of the world. So whatever biological spectrum we prepare of that area, it has to be compared with the normal biological spectrum. But this can only be done when you have calculated the percentage value of each life form, right? So now let us move towards the next slide, which is comparison of biological spectrum of desert with Ronquier's life form. So this this chart that you see, this graph that you see, is actually the world physiognomic flora which is given by this is Ronquier's normal biological spectrum. Now, what Ronquier did, he actually he actually calculated the all the kind of species that are present in the world. He did a very detailed study about the flora of the world, right, about all the plants present in the world, and he divided the flora present in the world into these 5 classes. So what he did, he actually found that in the world the percentage value of phanerophytes is 46 percentage, right? So these are the life forms and this is the percentage value. So when he plotted the graph, it was found that phanerophytes were 46 percentage in the world, chemophytes were 9%, hemicryptophytes 26%, cryptophytes 6%, and therophytes 13%. So he calculated the percentage value of each life form in the world. This is the distribution in world. So therefore this is called world physiognomic flora. That is, what is the percentage of each of the life form flora present in the world. Therefore, this is called Ronquier's normal biological spectrum. Now, as I told, after we have prepared our biological spectrum of the area, we have to compare that biological spectrum with the Ronquier's normal biological spectrum. Now, this graph that you see is the biological spectrum of a desert area. Right, this is a comparison of biological spectrum of desert area that we have studied with the biological spectrum of the world prepared by Ronquier. So we have to make this comparison. Right, so this is the biological spectrum of a desert. So as you can see that when the biological spectrum of desert was prepared, it was found that when we compare this with the world physiognomic flora, it was found that therophytes are maximum, which are 42%, right? And how did they find that these therophytes are maximum? Because they listed the plants present there, and based upon their characteristics they found that all of these plants fall under the therophytes life form, and therefore there is 42% value of plants that belongs to therophyte. So this is obvious that when there would be more therophytes, the area would be a desert, right? So higher value of therophytes indicates a desert habitat. So even if by looking by visualizing an area, sometimes you might not be able to identify that whether this is a desert or some other kind of habitat, but when you do this study of all the flora present there by physiognomic method, you will identify that what kind of habitat is present there. So as we did this study, it was found that therophytes are maximum 42% in desert, and therefore the higher value of therophytes indicates desert habitat. Whereas when we compare it with world flora, we see that all over the world only there is 13% therophytes present, but when I am going to study a particular desert area, the therophytes percentage is higher, right? So like this comparison of biological spectrum has to be done. This is the biological spectrum that we can prepare of an area that we have studied, which has to be compared with the standard Ronquier's normal biological spectrum, which gives us an idea of the habitat of that area, right? So this is actually physiognomic method. If you remember, we had seen that there are two things that we do in physiognomic method. First is that we record the life form and we calculate its percentage value. So that we already saw that we how to calculate the percentage value. And the second thing that we have to do in physiognomic method is to prepare the biological spectrum. So this is the biological spectrum that can be prepared. This is of a desert. Like this, a biological spectrum can be prepared of maybe a forest area, maybe a grassland, right? So biological spectrum has to be prepared, which has to be compared with the Ronkier's normal biological spectrum. So this gives us an idea of the habitat of that area, right? So this is about physiognomic method. Now this method, physiognomic method, has limited uses though, because it does not take into consideration ecological factors as biological disturbances. Sometimes we know that in a community there could be biological disturbances, that could be disturbances due to artificial things, maybe due to man, right? So these disturbances are not considered in this method. Now this we can understand with an example which is of Indo-Gangetic plains. Now the climate of Indo-Gangetic plains, actually it has to be forest. Okay, let me first explain you that what is Indo-Gangetic plain, because I think that you must not be knowing. So Indo-Gangetic plains actually is suitable for forest development, so phanerophytes should have been higher there. But let us first understand that what are Indo-Gangetic plains. For those who do not know, this part of India that you know, right, this part, the northern part, this is the south, this is the north, the northern states, all of these states, which are the states? This is Punjab, Haryana, Uttar Pradesh, Bihar, West Bengal. These are the states that fall under Indo-Gangetic plains. Now what is Indo-Gangetic plains? Indo-Gangetic plains means area which is the area of India, the states of India which are separated from Himalayas. So here actually Himalayan area comes. Okay, let us understand with this diagram. So these are the Himalayas and this is the Indo-Gangetic plain. So we know that from the Himalayas Ganga flows, the Ganga river flows, and so therefore all of the states that are present here is going to get Ganga water, right? Now we know that when water flows from the top, from mountains, it brings along with it all kinds of minerals also. So the Ganga brings all the minerals to these states, right? And therefore these states are actually very fertile because these are receiving minerals from the mountains. The soil of this area is, these states are very fertile. Here any kind of plants can be grown because the soil has all kinds of minerals, and therefore it is said here that this area is suitable for forest development, right? So therefore this is called Indo-Gangetic plain because Ganga flows from here, it comes from Himalayas and it gives water to all of these states, and these states have very fertile soil. Therefore it is suitable for forest development, and therefore phanerophytes should be higher in this area. Phanerophytes, we know these are tall trees, right? So such kind of trees, life form should be higher in this place because of the fertility of the soil. So basically what is Indo-Gangetic plain? This is Indo-Gangetic plain, all the states that come in this part. These the Indo-Gangetic plain actually separates the peninsula from the Himalaya. So peninsula is nothing but India. Peninsula is a place which is surrounded by water on three sides. So if you see, India is surrounded on three sides by water, and therefore India is called peninsula. And so Indo-Gangetic plain is area that separates the India, or we can name it as peninsula, from the Himalaya. So this Indo-Gangetic plain is separating our country from the Himalayas. So this is like a border that is formed. So these are plain regions, but these are receiving minerals due to the flowing Ganga river, and therefore the area there is fertile, and therefore it is very obvious that when there is fertile soil, there has to be forest development, and so therefore the phanerophytes should be higher there, right? But now see what is happening, that when the biological spectrum of this area was prepared, therophytes were found to be more. Right, so now we know what are therophytes. These are desert plants. So when the biological spectrum was prepared, it was found that therophytes were more, and the more percentage of therophytes is mainly due to biotic influences of man during agricultural practices, or maybe due to grazing effects, scrapping, etc. See what happens when man starts interfering. So what will happen, that the forest area starts getting cleared, because man has to perform agriculture activities. So they will clear the forest, and this will lead to the removal of forest patches, and this will lead to agricultural area, right? So in this way forest reduces, and second, there is grazing effect also. So there is more grassland now, and there are animals that are to be taken care of. So there is grazing effect also seen. That has also, now more the grazing, more the clearance of green patches, right? And therefore also there is loss of forest, and due to these practices there was found more of therophytes rather than phanerophytes. Though this area is very fertile, it was found in the biological spectrum that therophytes were more, right? So this is how biological disturbances can cause harm to a particular area, right? So this is about physiognomic method, where we have to actually make a biological spectrum of that area, which gives us an idea of the habitat of that area. Right, now we move towards the understanding of phytosociological method, which is the third method. So for that, first we need to know that what is phytosociology. So phytosociology is actually a branch of science which deals with plant communities, their composition, development, and relationship between species within them. So I think we can very simply understand this term with the term itself. Phyto means plants, and sociology means to socialize, right? So what we are doing in phytosociology, mainly we are trying to learn about the relationship between the species within them. Now see, plants also socialize, right? They also have a community. Species, all kind of organisms have community. So we are going to understand about the relationship between the species within them, then it is called phytosociology. So this is a branch of science which deals with plant communities, and what is its composition and development, and how is it related to all the species. So that is phytosociology. And therefore then phytosociological methods gives us detailed information on the composition, structure, species diversity, growth, trends of succession, other characteristics. So phytosociological method is a more used method to study the characters of plant community. As I told, floristic methods are obsolete, but phytosociological methods are still used um, to obtain the characters of community. Now here a very important term that we need to understand is sampling unit. So what is sampling unit? So the units that are selected for the purpose of sampling. When we have to perform sampling, when we have to take a sample of vegetation, we have to decide on a unit. So when we have to decide on a unit, so whatever unit that we are going to select to sample the vegetation, that is called the sampling unit. Right, so mainly three forms of sampling units are considered. These are area, line and point. These three are the sampling units that are considered in phytosociological method, and you will observe now that there are three phytosociological methods which are related to these sampling units. Right, so for now you remember that there are these three sampling units that are considered in phytosociological methods. These are area, line and point. So area and line, we know, has a definite area, right? So this is for suitable size of sampling. Now this has a size, right? So where will we take area and line? In places where we can consider size of the area, in such area we will take area and line as the sampling unit. But when we go into very thick forest, right, which have very long grassy and shrubby belts, it becomes actually difficult to have a definite area. Right, this is very obvious that when we move into a forest, the forest is very big, very thick, and it becomes very difficult for us to identify, to get a definite area of the forest. So we cannot move all along the forest and keep on performing size sampling. So in such cases we have to use the point as the sampling unit. We will understand this sampling units, you will understand more in the next slide. So now, as I told, on the basis of the nature of the sampling unit, there are three types of phytosociological methods that are used. These are Quadrat method, Transect method and point method. Right, so this, as I told, on the basis of the sampling unit, this bifurcation has been done. So in Quadrat method, the sampling unit is definite area. Remember, the one sampling unit was area. So in Quadrat method, the sampling unit is definite area. Now this area that we are talking about is taken in the form of a square, rectangle or a circle. So area could be taken, it could be in the form of a square shaped area, a rectangle shaped area or a circle shaped area can be considered for Quadrat method. So for example, this is a forest floor, right, the floor part of the forest, and if I want to study this with Quadrat method, so what I do, I keep a quadrat over this. This is a quadrat, right, and this is a square quadrat. As you can see, there are many small squares present. So what I'll do, that I will calculate the number of species in each of this square area, and on this basis I can get the character of the community. We will understand the Quadrat method in a bit more detail, but this is just an overview of Quadrat method, that in here area is used as a sampling unit, and the area could be square, rectangle or circle, and this is actually a quadrat that is used. I think you people might already be knowing what is a Quadrat method, but for we will have a little more understanding about this. For now we move towards what is transect method. So in transect method, the sampling unit is line of definite length. So as I told, the second type of sampling unit was line. So in transect method, the sampling unit is line which has to be of a definite length, that is, it will have a decided length. So this is transect method. This is a line. The white thing that you see is the line. It has segments, and you can also count the species present on the line, or you can keep a quadrat here and then calculate the species present in the quadrat. So this is transect method. And the third method is point method, where sampling unit is point. So as I told, there are three types of sampling unit. So we saw a method that is based on area sampling unit, we saw a method that is based on line sampling unit, and the third sampling unit is point, and point method is based on point as the sampling unit. So basically this is the instrument used in point method. We need not understand this in detail right now, but I'll be explaining you in the coming slides. So a very important thing that you need to know is, if you want to prepare a quadrat like this, it is not necessary that the quadrat should have a wooden frame, it should have these kind of mesh, right? We can also prepare quadrat with very simple materials. The three important materials that we might need is needle, strings, right, that is the thread, and hammer, right? So what we can do, I can identify for example, this is a forest area and I want to make a quadrat here with this material that I have. What I'll do, that I will keep one nail here, and I will try to fix one nail here, one nail here, and one nail here, right? I do not have a wooden frame, so I want to make a quadrat like this wooden frame. So I'll fix nails on the four corner, and then I will take a thread and I'll start from here and tie it here, start from here and tie it here, and like this I can complete a square with a thread. And then in there also I can make quadrats with the help of threads. So this is the very basic things that are needed to make a quadrat. Now let us understand a bit more about quadrat method. So as I told, here the sampling unit is not a quadrat, it is a area. So it could be square, rectangle, or a circle. Now this quadrat method, there are mainly four types of quadrat method that are used. These are list quadrat, list count quadrat, chart quadrat, and experimental permanent quadrat. So very easy actually. List quadrat, as the name itself suggests, here in this quadrat we are only going to list down the species that are present in the area. So whenever we are going to study any area, what we will do in list quadrat, we will only list down the species. Say for example, in an area we found that there is Cynodon dactylon present. Okay, I list it down. Then I list down the second species, then I list down the third species, then I list down the fourth species, and like this I can keep on listing the species present there. Then this is called list quadrat method, where only I give the list of the species. Now the second method is list count method. What is done in list count method? The name itself suggests that apart from listing, we are also going to count the species. So along with listing, numerical counts are also made, right? So what is done in this? So as we saw, we had already listed the species, but now what we will do, we will count the number of species. So how many cynodon dactylons are present? 5 are present. How many rose plant are present? 2 are present. Okay, how many cosmos species is present? 1 is present. So this we are also taking a count of the species, then it is called list count quadrat. Then chart quadrat is a method where we show the cover of the species over a graph paper. Here instrument called pantograph is used. So pantograph is used to identify the cover of the species in an area, but pantograph is a big instrument. This instrument actually gives us detail to scale, growth and distribution in each species is also recorded. And what actually pantograph does is it will actually scale down the growth to a very small area. So pantograph is actually a tracing device. So it is a tracing device that helps in drawing to identify cover. Right? What is a pantograph? It is a tracing device. So with that you can trace the area, the cover of the plant, right? How much area or how much cover the plant is taking. So the pantographs helps in tracing that. So actually what pantograph does, it reduces the drawing to scale onto graph paper. So when we actually see in a forest, it is a huge cover area, right? But what if I want to draw it on a paper, right? So pantograph actually reduces the drawing scale, and so that you can draw the cover on a graph paper. So like this you can draw the cover of the species. So for example, this is the area covered by species 1. So for us species 1 is cynodon dactylon. So I can show a patch of species of species 1. Similarly species 2 is present here, species 3 is present here, species 4 is present here. Likewise species 5, which is Catharanthus, is present in 2 patches, and in this way chart quadrat can be prepared to identify the cover of the species. So actually I have not kept here the diagram of pantograph because I did not get properly I did not get an image to keep here. So maybe you can Google pantograph for studying plant community, and you will identify, you will be able to see what a pantograph looks like, right? And the fourth is experimental permanent quadrat, which we are going to see in the next slide. So experimental permanent quadrat is, as the name suggests, that this is a permanent quadrat, that is we are not going to disturb the quadrat, and this is mainly for experimental purpose. So what we do in an experimental permanent quadrat, the chart quadrat is left undisturbed and the area is studied for vegetational changes over long period of time. So what we will do, that whatever quadrat that we have taken, we will keep it undisturbed in that area, right? So we are not going to disturb that area, and the quadrat will be kept as it is. So what will happen, that this is done actually for a very long period of time for experimental purpose. So if you keep the quadrat there in at the same place, what will happen, that you can see the vegetational changes taking place over there over years, right? So a plant might be small when you would have kept the quadrat there, but after 5 years if you see that there might be change in that plant and it must have grown larger, right? and there might be even other plants there which you may not have seen when you had kept the quadrat, right? So this is a permanent quadrat where you study the vegetational changes. This study is the periodic changes in community. So we know with time the community keeps on changes, and these changes can be studied with the help of experimental quadrat. This is also useful in successional studies. Now you already know what is succession, right? The changes that takes place continuously, this is succession, and we can also study successional changes with this kind of quadrat. So 4 things we need to do for experimental permanent quadrat. First is I need to determine the size of the quadrat. So I need to know that what is the size of the quadrat that has to be calculated. Then second is to determine minimum number of quadrats, that is how many quadrats do I have to keep. Do I have to keep 3 quadrats? Do I have to keep 10 quadrats? So I need to decide on a minimum number, and then I need to keep on recording the species present there, and then the calculation of frequency, abundance and density of that species is done. So to determine the size of quadrat, there is a method used which is called species area curve method. By the help of species area curve method, the size of the quadrat is determined. Now what does a species area curve method tells? It actually gives us a relationship between area of a habitat and the number of species. So what is the area of the habitat that we are going to study? For example, if I am going to study a desert habitat, what is its total area, right? We have to see that. And then what are the number of species present there, that also we have to calculate. When we have these 2 things, right, so then we can decide on the size of the quadrat. So this is species area curve method, and this method actually estimates the minimum size of a quadrat which is necessary to characterize a community. So some areas might need small quadrat, some area might need large quadrats, right? So it depends upon the type of area and the number of species which are present, and based on that we can determine the size of the quadrat. Then we need to determine the minimum number of quadrats to be laid down. Now this depends upon the size of the area and number of the species again. So some areas will have more number of species, so then we need to keep sometimes more number of quadrats, sometimes 10 to 15 quadrats. Some area will have less number of species, so keeping only 1 quadrat might also be enough. So that depends upon the area and number of species. And then we have to keep on recording the species. So how we do? We list the species and we count the number of species, and then we can calculate its frequency, abundance and density, right? So now you might be thinking that how are we going to calculate frequency, abundance and density? So these how are we going to calculate is not the concerned topic right now, because that is part of the characters of community which I'll be explaining in later videos. For now you need to remember that whatever species we are recording, its frequency, density and abundance has to be calculated. So after we determine the frequency, density and abundance, we can calculate the frequency percentage of each species. Now I think now you already know how to calculate percentage, right? And this frequency percentage that we calculate is distributed among Ronquier's 5 frequency classes. Okay, so let us in a very brief understand what is frequency, because this is important, only then you will understand the Ronquier's 5 frequency classes. So frequency is what is the occurrence of a species in an area. For example, some species might be present in a large number. Say for example, in an area there is neem tree present. So how much number is that neem tree present? Is it repeating again and again? So that is frequency. So some species would have less frequency, some species would have more frequency, right? So in simple language, what is the spread of that species? So से फॉर एग्जाम्पल कोई एरिया में नीम ट्री प्रेजेंट है, तो उसके कितने नंबर्स है? क्या वो बार बार रिपीट हो रहा है? क्या उसकी फ्रीक्वेंसी ज़्यादा है, मतलब उसका परसेंटेज ज़्यादा है? हो सकता है कोई एरिया में नीम ट्री ज़्यादा प्रेजेंट हो, राइट, और दूसरे प्लांट्स जो है वो कम प्रेजेंट हो, तो वो दूसरे प्लांट्स की फ्रीक्वेंसी कम हुई और नीम ट्री की फ्रीक्वेंसी ज़्यादा हुई. मतलब वो कितना प्रेजेंट है, उसका कितना अकरेंस है एक एरिया में, उसके कितने नंबर है एक एरिया में, क्या वो ज़्यादा है नंबर, क्या वो कम है नंबर, उसके बेसिस पे हम फ्रीक्वेंसी परसेंटेज कैलकुलेट कर सकते हैं. सो Ronquier actually gave 5 frequency classes. What Ronquier told, that all those species that will have 0 to 20% frequency, right, will fall under class A. 0 to 20% frequency मतलब कि उस पर्टिकुलर स्पीशीज़ की फ्रीक्वेंसी रेट बहुत कम है, मतलब it is not present in a larger number. So these are species that are not present in larger number in any area. So these falls under class A. Now species that have 21 to 40% frequency falls in class B, with 41 to 60% frequency falls in class C, 61 to 80% falls in class D, and 81 to 100% falls in class E. So understand this table. See what does it mean to be in class E. This means that a species which is having maximum frequency 81 to 100% is class E, right? This means that what kind of species will come in this part? Species that are very vigorous, and species that is having very good spread, that are having very good numbers in an area, will be included under class E, right? So again taking the same example as I told, for example एक area है, right, and in that area say there are 100 species. Out of that 100 species, 80 species are neem tree itself. So it means that the neem tree will come under the frequency class E, because its percentage, if we go to calculate, it will come between 81 to 100, because the number of neem trees is maximum in that area, right? So this means that the community there is a homogeneous community. Homogeneous means a community that is not having much diversity, a uniform kind of community, because all the species present there are of this almost same kind. So this having a E value of a community means that the community will have homogeneous kind of arrangement, where the community is not having different kinds of plant, but there is only one kind of plant present again and again, right? So higher the E value, higher the presence of E value will show that it is a homogeneous community. Now there is another term that you need to understand is heterogeneous community. How can we say that a community will be heterogeneous when there would be more B, C and D value, right? Which means that there are some plants that have frequency of 21 to 40%. So in an area some plants have this frequency, some plants have this frequency and some plants have this frequency. So there are different kinds of plants present of different frequency. This means that this would be a heterogeneous community. So when a community has B, C and D value higher, so it would mean that it will have a heterogeneous community, right? I hope that this is clear with you, because we are going to understand this in the next slide in a bit more detail. And if you do not understand still, you can always ask me. I'll make you understand this. So as I told that Ronquier has given 5 frequency classes. Now whatever species we have recorded in a community, we have to find out its value in percentage, right? So we need to get the percentage of total species for each of the 5 frequency classes, and this will give us a frequency diagram that we can prepare. So see, this is Ronquier's normal frequency diagram. This is the diagram given by Ronquier, right? So we just saw that he has given 5 frequency classes A, B, C, D and E. So these are the 5 frequency classes, and this is the percentage of total number of species, right? So this is given by Ronquier. What he has shown that plants that belong to frequency class A are 53%. So 53% plants belong to frequency class A, 14% species belong to frequency class B, 9% belongs to frequency class C, 8% belongs to frequency class D, 16% of species belong to frequency class E, right? So this is normal frequency diagram given by Ronquier. Now this is a frequency diagram for grassland for our understanding, for our example, so that you can understand that how this frequency class becomes useful. So as you can see that here B, C and D value is higher. See, in Ronquier's normal frequency diagram, B, C and D value are less, but here B value is 20, here in Ronquier's it is 14. Here C value is 16, in Ronquier's it is 9. Here D value is 16, in Ronquier's it is 8. So when there is higher B, C and D value in comparison to Ronquier's normal frequency diagram, this means that this is a heterogeneous community. So in a grassland, basically you can find heterogeneous community where there would be different kinds of plants present actually. And a higher E value, as I told, will indicate homogeneous community. What is E class? It has 81 to 100% frequency, which means that one kind of plant is present repeatedly in a large number, right? And this is why the area is not having any different kinds of plants, and therefore this would be a homogeneous community, right? So when there would be higher E value, then it would be homogeneous community. Here E value is not that higher, and therefore this is not a homogeneous community, but B, C, D value are much higher in comparison to Ronquier's normal frequency diagram, and therefore this would be a heterogeneous community, right? So this is it about quadrat method, experimental quadrat method. Now we will understand transect method. So as I told, there is this quadrat method, transect method and point method. So quadrat method is completed. Now we move towards transect method. So here the sampling unit is line, right? So what will happen here, sampling strip is extended across stand or several stands of vegetations. We will understand this. Transect method are basically of two types, line transect and belt transect. So for those who do not know what is transect, transect is actually a path, a kind of path, straight path, and on that path whatever things come, that has to be counted, right? So let us understand this with line transect. Line transect means that we are taking only a thin line, and this is mainly used for grassland. So this figure that you see is an example of line transect. So this is one line that we have considered, right? So this is a definite line. So this is a kind of path, a straight path. Now whatever species, whatever plant species that will come over this path will be calculated, right? So I will count the species that will fall over this line, and the species that will not fall over this line is not counted. This is one kind of one way of measuring transect, one way of methoding the calculating the vegetation. So let us understand this with a forest diagram. So this is a forest floor. I have taken one thread from this side to this side. So this is a line transect. Now what I can do, I can also keep a quadrat in one segment, and in that segment whatever species come I will list and count, right? So I can keep a quadrat here, then I'll calculate species present here, and I can move this quadrat to the next segment, and I can also calculate the species present there. So this is a line transect. So either I can calculate the species present on the line, or I can calculate the species present in this quadrat, and this quadrat is in combination with the line, right? Now what is belt transect? When two lines are taken parallel, then it forms a belt, and therefore it is called a belt transect. Here belt of suitable length is used for forest. So what is done actually, that the transect here is divided into segments of suitable length. Let us understand this with an example of ecotone. This method is mainly useful to analyze the vegetation changing in its composition through an ecotone. So for this you need to know what is an ecotone. So ecotone is an area where two different communities meet, right? That is called ecotone. So we can understand with this example. Let me zoom it. See, this is an ecotone. Here it is tundra kind of climate. Tundra means cold climate, and here it is grassland, right? So the vegetation that would be present in tundra would be different, and the vegetation that would be present in grassland would be different. This middle portion, the line that is the attachment between the two communities, called ecotone. So the communities that fall in this area is called ecotone community, right? We can also understand this with a second example. So this is also a ecotone. See, this is a grassland and this is a forest. So the community of grassland will be different and the community of forest would be different, and this is called the ecotone. and we can also understand this with this diagram, this photograph, right? So this is also an ecotone. The community of aquatic area would be different, and the community of the river bank area would be different, and this is also a ecotone. So whenever we have to understand ecotone community, mainly during such times belt transect is used. So what is done in belt transect? This is one line, then it becomes line transect. Then I take another line, so this looks like a belt, and in the belt, so now you can see that there are both kinds of community present in the belt, the forest community also and the grassland community also. And I can now divide this into different segments, one segment of one quadrat, second segment of second quadrat. Like that I can keep on making different segments and different quadrats. So therefore it is written that the transect here is divided into segments of suitable length, right? And each segment may be taken as a quadrat. So every segment may be taken as a quadrat, and the species falling there would be calculated. So again the frequency density value of each is calculated, and in this way transect method can be useful. Now let us understand the point method. So here the sampling unit is point, and here point frame is used. So what is a point frame? This is actually a wooden frame, and there are 10 movable pins present which are inserted in a wooden frame. So let us understand this with this example. This kind of structure is used. This is a point frame. This part that you see is made of wood, and these are actually pins. Usually 10 movable pins are present. So you can also understand with this diagram. See, this is a point frame, this is wooden area, and this long needle like thing that you see are long pins. So in the frame there are holes, and these pins are inserted in the holes, right? So pins are laid down at random in the area. So whatever area you are studying, you keep this frame there, so the pins will fall over certain plants, right? So plants that are hit by pins are recorded. Now see, this is important. How plants are recorded in point method? So this is for example a point frame, right? And the pin has is hitting one plant. So for example, this pin has hit this plant, then this plant is counted. So when a pin it actually hits whatever plant, that plant is calculated. Now see, this pin is not hitting any plant, so this will be counted as a miss, so nothing is present here. Similarly, this pin is hitting a plant, therefore it will be counted as one species. Again this will be counted as one species, but now this pin is not hitting the plant, and therefore it will be counted as miss, right? So there might be many species present, but it is obvious that the pin is not going to hit every species, and so only those species that are hit by the pin will be recorded for the purpose of study, right? So the plants hit by pins are recorded, and then again the frequency percentage of each species is calculated. So in this way point method can also be used. So one hit of pin taken by a species is considered its occurrence. So जब जो पिन जिस प्लांट को हिट करता है उसी प्लांट को कंसिडर करते हैं, ओनली दैट प्लांट इज considered as the plant is present. Though there might be other plants also present, but if it is not touched by the pin, then it is not considered. Right, so this is a point method. This is usually used in forest, because in forest we cannot identify a specific size, because forest is very big. So we use this point method where randomly area is considered. Right, so I hope that this is now very much clear with you. But if in any case you people have any doubt, please feel free to ask me. I'll make it clear for you. Right, now also for density calculation, random pair method and point centered method are used, but we are not going to understand this because this is not a part of your study as of now. So with this we come to the end of the session, and please, if you have any doubt, please feel free to ask. Right, and if you feel that the content of my channel is useful, then please don't forget to subscribe and hit the bell icon. Thank you.